Okay, hi you guys. This is the 18.1 little lecture here I'm going to go through. Just a heads up, it was brought to my attention by a couple of students that emailed me that the guided reading I gave you says chapter 18, but it's actually chapter 19 information. So I'm not sure quite how that happened, so I didn't catch it. So don't worry about doing that over the weekend. Yay. I will post it. You will eventually have a chapter 18 guided reading, so I will post it to my website if you want to use the 4-day weekend for time to work on that and kind of work ahead. That's fine, but it won't be due on Monday. So chapter 18 is all about viruses and bacteria, and this first section is all about viruses. So the overview here, viruses weren't detected for a long time. You can't see them underneath the microscope, and so this first part of the section goes through and gives you a little bit of background about the structure and how they figured it out, and then about how viruses reproduce. All right, so really the discovery of viruses came with this tobacco mosaic disease, which is a disease that affected tobacco plants. And basically this virus that nobody could see made the plant's leaves appear splotchy. And so that's the mosaic color, and it stunted the growth. So you could obviously see something was wrong with it. So they studied it and couldn't figure out what was wrong with it. They couldn't grow it on a culture medium in a petri dish like you could bacteria. You couldn't see it underneath the microscope. So hence the second bullet, they hypothesized that a particle smaller than bacteria caused the disease, since they couldn't see it. And then this guy named Stanley confirmed this by crystallizing the particle, and now they call that the tobacco mosaic virus, or TMV for short. And so that's a little of the history. To give you an idea of size, let me put this picture up here. This picture is figure 18.2. I didn't put it in your packet, but it kind of gives you an idea that this little guy here is a bacteria. This is a virus, and this part right here is the animal cell. And so you notice in this picture that you can only see part of the animal cell. Here's the animal cell nucleus right here. So the animal cell is very large. Next comes the bacterium, which are still very way larger than viruses, but compared to the animal cell, very, very small. And then the virus is smallest yet. And so that's why they had trouble seeing and visualizing them and figuring it out. So viruses are a little bit different. Viruses are not cells. They are these very small and obviously infectious particles, and basically they have two parts. The genetic material, which is the nucleic acid, and notice I put here in parentheses behind it that could be DNA or RNA. So there are DNA and RNA viruses, and then they're enclosed in a protein coat. And in some cases, like the viruses that infect animals that we'll talk about here today, they have an envelope around them. So let's look at the genome. So genomes, we set up here that they could be DNA or RNA. So DNA, there's different kinds of DNA viruses. If the DNA is the genetic material, they could be double or single-stranded. We have double-stranded DNA, so viruses are a little bit different that way. And if they have RNA, it also can be double or single-stranded. So far what we've talked about with RNA is that they're single-stranded, but here they can be double-stranded as well. So viruses are classified based upon their genetic material. So you can hear the words DNA virus or RNA virus, and that is based upon what their genetic material is. So around their genetic material, they have a capsid and sometimes an envelope. So a capsid is a protein shell that contains or encloses the viral genome, whether or not it be DNA or RNA. And it can have lots of different structures and appearances. So that's what the next picture shows you, is the appearances or structures of the various types of viral capsids enclosing their genetic material. So you can see here, we just talked about the tobacco mosaic virus, and that's letter A here. This is an actual real picture of it, and this is a computer-generated picture. So it looks like a cylinder. Around here is, it says, the capsomere of the capsid. So this is the protein coat, and enclosed in there is the RNA. So if I were to draw this like this, and blue is the protein coat, and inside you have the RNA, which is the genetic material. So that has one particular structure. This is an adenovirus. This is a little bit different. It has a different shape, you can see. But these little balls here are still the proteins. This is the capsid, and these have glycoproteins. We're going to see later that these guys can help bind to the cell that it infects. And this happens to be a DNA virus because inside that capsid is the DNA, which is the genetic material. Influenza virus next, you can see here, now notice here it has, the capsid is pointing to these little cylinder-shaped things, and then around it is this membranous envelope with a glycoprotein. So what does that mean? If we draw a little bit on the side here, so what they have is this envelope around it and these glycoproteins sticking out. This is different than the one over here, like the tobacco mosaic virus does not have a membranous envelope like the influenza virus does. And so so influenza virus infects animals, and so we're going to see a lot of animal cells have this membranous envelope. And then inside of that, all these little cylinder-shaped things are the capsids. So inside of that envelope, you have in green here, these are the capsids. Capsids, so that's the capsid. The capsid, remember, is that protein coat. And then the capsid always surrounds the genetic material. In this case here, notice here for the influenza virus, that genetic material is RNA. So you have RNA inside here. And so there are many capsids around an envelope. And so that's a little bit different than the other two that we talked about here. And then the last one is the bacteriophage, which we've talked about before, is a virus that infects bacteria and has this unique shape here that we looked at previously in class. And so the capsid has this head part and then has the DNA inside of it. And then it has this tail part with these little tail fibers that, remember, can land on the bacteria and inject their DNA in it. So all kinds of different structures, but have some similarities between them. So that's what we get at here as we look into the bullets here. So some viruses have structures that should say called membranous envelopes that help them infect the host. So up here, that was the example of the influenza virus. Influenza virus. And then they surround the capsids, that's what we just drew, of the influenza viruses and other viruses that infect animals. The viral envelopes, which are derived from the host cell's membrane, we'll look at that in just a second here, contain a combination of viral and host cell molecules. So their host cell is the cell that they're going to infect. And so there's a combination of those. And when we look at how they reproduce, that'll make a little bit more sense. And then that last one, the bacteriophages, are also called phages and are viruses that solely infect bacteria. They have the most complex capsids, the unique little shape there, found among viruses. And they have the capsid head that encloses their DNA, and then their tail piece with the fibers are made out of protein. And that tail piece, remember, attaches to the host, which is the bacteria, and injects the DNA inside. And so that's how they work. So that's just a brief overview of the structure of the viruses. Now we're going to look at how viruses reproduce. And the thing about viruses is they cannot reproduce on their own. So viruses are what we call obligate intracellular parasites. That means that they can only reproduce within a host cell, so they can't reproduce on their own. They have to use the host cell's kind of machinery. And each virus has a host cell range, which is a limited number of host cells that it can infect. And so not every virus can infect everything. So a virus that infects a specific type of plant may not infect all plants and may not infect any animals. And so it's very, very specific in terms of the hosts that it infects. And so when they can't reproduce on their own, what they're using is the host cells' enzymes. They're using the ribosomes and other molecules in the host cell to make, it says progeny viruses. That means new viruses. Basically, long story short is it's going to use the host cell as a virus-producing factory and produce lots of different viruses. So this picture gives you a general overview of what that is. This is figure 18.5 in your textbook. And notice it says a simplified viral reproductive cycle. So this is very simplified to give you an overview. And then we're going to look at two different types of viruses, bacteriophages and then the viruses that infect animals, and give you some specifics. So this is an overview. So here you have in red the capsid, which is that protein coat, and in blue the DNA. So when the virus infects its host cell, so this little guy here is its host cell, notice the virus kind of comes apart. So this is a DNA virus. So here's the viral double-stranded DNA. So it's a double-stranded virus, and here's the capsids. So what happens is the virus uses the host cell to use the host cell uses the viral DNA to make new viral proteins and to reproduce its DNA. So notice here there's two different pathways. So host cells have their own enzymes, DNA polymerase, and so on, to make DNA. That's DNA replication that we just talked about. So when the virus goes into the cell, it uses the host cell, and the host cell doesn't know any better and starts replicating the viral DNA just like it does its own DNA. And so now this is many copies of the viral DNA. So that's part one. Part two is the second thing that a host cell, a normal cell, will do is use the DNA to make proteins, and that's protein synthesis. So what we see going on this side is that the DNA is used to make mRNA. So here's your mRNA. And your mRNA, that's transcription, then your mRNA is used to make proteins, that's translation. And since you're doing this with the viral DNA, what you're making is viral proteins. So that's what this is showing you here. You're making new capsid proteins. So number two here, the host cell enzymes transcribe the viral DNA into mRNA. That's this point here. And then from here to here, that's translation. The mRNA will go to the ribosome of the host cell and be used to make the proteins, the viral capsid proteins. Then what happens is they come together. And so here's your double-stranded DNA. Here are your capsids, and new viruses are made, and these viruses leave the host cell, and now you have more than the one that you started out with. And these guys, what can they do? They can go attack other host cells. And so they've used this host cell. They haven't killed it, and they've used it and its machinery, its ribosomes and molecules and so on, and enzymes to make new viruses. Viruses have to have a host cell to do this. So that's an overview. So let's look at one type of virus next. So the reproductive cycles of a phage, which is, remember, short for a bacteriophage. These guys only infect bacteria. All right, they're the best understood of all viruses. The most research has been done on them, and they're unique in that there's two reproductive mechanisms called the lytic cycle and lysogenic cycle. And so let's look at both of these separately. So the lytic cycle, it culminates in the death of the host cell. So unlike up here where the host cell stayed alive, this one's going to cause the host cell to die. But it has the same outcome in terms of the number of viruses. It produces new phages and digests the host cell wall, releasing the progeny viruses, which are the new viruses. this type of virus, this type of bacteriophage that reproduces only by this cycle is called a virulent phage. It kills its host cell but makes many new cells, or not new cells, new viruses. And bacteria actually have defenses against these types of phages, including restriction enzymes. Restriction enzymes are special enzymes that recognize and cut up certain phage DNA. So let's look at a picture that illustrates this. Okay, so this right here is the lytic cycle of the bacteriophage. So what we have is your bacteriophage that, remember, is the protein coat, that's the capsid, with these tail fibers, remember? And remember, they land, this is the bacteria cell, so the bacteria cell is the host cell. And so inside the bacteriophage, you have its DNA, so that's in green here, the DNA. So what happens is this DNA is going to be inserted into the host cell. All right, so here's your DNA going to be inserted into the host cell. And we can see that again in this picture here. The DNA is being inserted into the host cell. Now notice what's happening to the host cell DNA. The host cell DNA, remember, bacteria's DNA is one big circular chromosome. So that's what we see here, one big circular chromosome. And notice here that it's being cut up. All right, so as the phage enters and it degrades the host cell's DNA. Then what it does is, as that phage DNA goes in, the phage DNA is used to, as we just learned, to the host cell, takes that DNA and makes more DNA of the viral DNA. And so that's what we see here, all kinds of viral DNA being made. And so it replicates the viral DNA, but then it also uses that DNA to make more proteins. So what are the proteins? These are the capsid proteins. So you can see here around here, you have your capsids being made, you have your tails being made. These are the tail fibers. So it's making all kinds of new viral proteins, and then it assembles them. So you can see inside of here that, like, for instance, here's a full capsid with the tail fibers here, and inside you have the viral DNA. And then so you get this this bacteria chock full of these viruses, and then the viruses also make an enzyme that lyses or breaks the cell wall of the bacteria. So that's what you see here, that they're actually, looks like it's kind of exploding, which means that all these new viruses now can go out and infect, and these guys will go infect new bacteria. Okay, and so the host cell has died, and the virus has made many new viruses using that host cell. Okay, so this picture right here just shows you the assembly. So when they're going together, all the different parts of it, the capsid, the tails, the tail fibers, and so on and so forth. And that is the lytic cycle of bacteriophages. There's one other cycle of bacteriophages, and that's called the lysogenic. The lysogenic doesn't destroy it. It replicates the phage genome without destroying the host. So, so let's go over, I'm going to come back to this fill in the blank here, and let's go over the parts of it first. So I'll go to the next page here where you have this picture. This is the lysogenic cycle. And what I want to point out here is this shows you both in combination with one another. So this right here is the lytic cycle, or sometimes called lytic cycle, and lysogenic cycle. And so we have these two. This is what we just talked about. So let's just look at this here. Here's the phage DNA, the injects in there, and so normally then in the lytic cycle, it'll break the bacterial chromosome up and make new viruses, and then the host cell will explode and all the new phages will go and infect another cell. So that's what we just looked at. But I want to point out that, so for the lysogenic cycle, we start with the same process here, where the phage is injecting its DNA. So here's the DNA inside, and it's being injected. This is the bacteria's chromosome, all right? So then we go to this next part here. So the phage separates here. Here's the the virus's DNA. And normally in the lytic cycle, it'll break up the bacterial chromosome and go in this way. So that is one pathway. So that's one pathway. But another pathway is here. So we can go in either way. So the lytic cycle, or we can enter into, or the bacteria host cell will enter into the lysogenic cycle. When the lysogenic cycle is entered, what happens is, instead of destroying the bacteria's DNA, the viral DNA actually becomes incorporated into there. So that's called a prophage. So the phage DNA integrates into the bacterial chromosome, becoming a prophage. And then it just kind of stays in there. It's like silent for a while. So then the bacteria is just going along its merry way, living its life and doing what bacteria do. And one of the things that bacteria do is they replicate, and they replicate very quickly. They divide. So when a bacteria cell divides, it makes a copy of its circular chromosome and then divides. And so when it makes a copy of it, it treats the viral DNA just like the bacteria's DNA. So therefore, the new DNA both have the prophage in it. And so this bacteria cell is going to, you can see here, it's going to indent and divide right here. And then each new cell is going to look like this. This is one of the daughter cells with the prophage in it. And it keeps on doing that. So what's going to happen is, over time, you're going to have millions of bacteria that have this viral DNA in it. And then occasionally, notice here it says occasionally, a prophage exits the bacterial chromosome and initiates the lytic cycle. So then look at here, the prophage pops out, bacterial chromosome is back to normal, and it enters the lytic cycle and now uses that to make lots of viruses, and then it kills the host cell. But in the meantime, you've gotten, by doing this lysogenic cycle, you've gotten millions of bacteria that have this viral DNA, and at any given time, that DNA can pop out and they can release new viruses through the lytic cycle. So that is a way for the bacteriophages to get lots of cells with the DNA, and at any given time, each one of those cells can do this and get lots of them viruses. So the lysogenic cycle by itself doesn't produce new viruses. It just reproduces cells that have the viral DNA that can make new viruses by entering the lytic cycle. And notice it says here what triggers that. Certain factors. So this could be environmental factors like chemicals and things like that in the environment that can trigger it to, and the bacteria cell and the viral DNA to leave the lysogenic cycle and enter the lytic cycle. And so that's unique about bacteria. So bacteria have evolved some mechanisms to help protect themselves against viral viruses like the bacteriophages. So for instance, I mentioned that they have restriction enzymes. So restriction enzymes cut up foreign DNA. So some bacteria, if they have the right restriction enzymes, can cut up the foreign DNA and prevent themselves from going into any of these cycles. The thing is that some bacteria cells may have it and some that don't, and so the ones that do will live on and pass those genes on to make those restriction enzymes through that whole process of natural selection. And so bacteria cells that have the appropriate restriction enzymes proliferate, they divide, and so that's how bacteria kind of have lived on in the presence of these viruses that infect them. Okay. All right, so then the last little bit is the reproductive cycle of Okay, so we're going to do animal viruses, but I just got a pop-up message on my iPad here that I'm recording that my storage is almost full. So I'm going to stop this video so I can take care of that. And so I'm going to put a second installment for the last few pages for animal viruses. So just click on that to watch the rest of it. Sorry, guys.